So as a background of transcription, RNTPs are the building blocks of transcription, and they will make up the RNA. Phosphodiester bonds will link those RNTPs together, and RNA polymerase is the enzyme that will synthesize that RNA. To start off, RNA polymerase binds to the promoter regions of the -10 and -35 zones on the DNA to start transcription. The RNA polymerase is made of parts, and that is what is the hollow enzyme, and it's all the parts plus the sigma factor, and that sigma factor is what actually recognizes the -10, -35, and binds to the DNA molecule to start transcription. Once bound, the RNA polymerase starts transcription, and the +1 area is the first transcribed nucleotide. Once the DNA molecules are separated, the RNA polymerase starts adding RNTPs in the 5' to 3' direction. It uses the template strand to synthesize the RNA, so it adds those conjugate RNTPs to the correct DNA bases. The coding strand is essentially identical to the synthesized DNA, but it has thymine instead of uracil, and it is not used as the template. An inverted repeat is coded into the DNA, where a sequence of bases has the same conjugates later in the sequence. So this repeat will allow for base pairing within that strand, and this will cause termination of transcription. The loop causes the RNA polymerase to pause, and the long sequence of AU pairing between the RNA and DNA after the loop promotes the RNA to dissociate from the DNA. Once the RNA dissociates from the DNA, it will terminate transcription. Once terminated, the RNA will go to the ribosome to be made into proteins. So let's say that the -10 box is deleted from a gene that codes for an enzyme that metabolizes sugar. If this happens, then the sigma factor of the RNA polymerase will not be able to recognize and bind to the promoter region of the DNA strand. And if the RNA polymerase cannot bind, it will not transcribe the DNA into RNA. The RNA will then not be made into proteins from the ribosome, and without the proteins made from this gene, the organism will no longer be able to metabolize sugar like it normally would.